All right, so let's get started with day 3 of this workshop, RNA-seq I. So in the last 2 days, we have gone through pretty much like the majority of the part where we start with the raw read files in FASTQ format, and then we go through quality controls. We know how to filter the read for only keep the high quality ones, and then we know how to pick the reference genome, how to index them, and we of course did the alignment, which is the most time-consuming process of the entire process. And then we tallied the per gene read count, so we end up getting that read count table for a couple of the sample data we have tried. And then I think some of us have gone through the pipeline using the mouse liver data. I hope it's working fine now. I don't know what happened after the class yesterday. So if you haven't got a chance to try, you're free to try anytime. The code is going to be up on the web. So if you want to save it for future references, you can export or print as a PDF, and it should enable you to kind of still copy the code as you go. So for today, we want to kind of get into the application side, which is like, okay, can we actually find out potential public data that are available for our own research? How do we do that? Again, the Hoffman tool is going to be a good friend for that. It's pretty fast in downloading and storing and processing all the data. Because for all those bulk and seq data, it's quite big, especially when you have multiple samples. And then we want to go through a little bit about how we deal with the count matrices we have generated from the pipeline, especially when you have multiple samples and you want to compare between samples. Let's say the same gene, can we actually compare it in a fair and kind of well-controlled way? Especially some of the samples will have more sequencing depths and they have more mRNA to begin with. We have to account for this kind of biases in sample quality. And then we want to go a bit into the visualization step. So now you have this big matrix, they're all numbers. How do we actually generate maybe potential publishable figures so that to best represent the data set you are interested in? So that's for today, and we would like to get started with the first part, which is going through public RNA-seq analysis and trying to find the data that are available. So many journals now require you to upload all the raw sequences if you are doing a next-gen, high throughput experiment. So that includes RNA-seq, both bulk and a single-cell data should be, deposited in this gene expression omnibus. So this is kind of a one-stop kind of shopping place for all the the gene expression data. And then besides gene expression, I think nowadays all the ChIP-seq data has to be deposited, and then some of the protein data are now also getting deposited. Especially we're now getting to the age of single-cell proteomics. So it's quite a really good resources for the whole community. Especially we want to make sure that all the results are reproducible. So having that data available is super helpful. So we want to kind of use one example to demonstrate that, how to find the data set we're interested in and how we can actually download the data on Hoffman. So the example we're using is this entry with the session number GSE 117673. So when you search for this number, so this number has to be reported in the publication. So you can kind of just Google that, and it will take you to this page where it has kind of list of information. For example, the title of the study, who generated the data, and what kind of data is there. So if you kind of just look through the website, you can get quite a lot of information. For example, what sequencing platform was used. So in this case, they use the Illumina Hi-C 4000 for 2 species. So we are looking at the soybean data here. So there are, kind of a lab-grown version, which is glycine X, and then there's a Y-type glycine soya. So, it has this very basic information for you. And then there's a list of samples. So those are individual sequencing samples. So that number means just, how large the sample size we're dealing with. In this case, they have uploaded 4,003 samples. So we can have a look at individual one of them. So what we do is...
What was the platform again? Just what the platform that was used to do
for the sequencing machine.
Okay.
Sometimes, it does have different biases. It can be high-C sequences, or it can be, let's say, BGI. Some other machines are also used for next-gen sequencing. So if we go into the SRA run selector, that's where actually you get the raw data. So that's the fastq.gz files that we started with in the day one. So if you click into the SRA run selector, this is the page you will get to. And here is kind of, a general overview of all the data. So you can choose to download them and you can see how big they are. So for the 1,003 samples, we are downloading about like 1.7 terabytes of just raw reads, basically. But you don't need to download all of them. If you're only interested in some samples, you can select, kind of there's a toggle switch here to select a subset of the sample. And there are a lot of those big tables containing information about individual samples. For example, which species are they sample for? Is the lab-grown version or the wild type of soybean? And another condition will be, is there different biological variables, for example, different genotype or photoperiodicity? So they have a long day, short day condition, or they may be sampled from different time. So those are the information to kind of guide you as what samples I should focus on instead of downloading all 1,000 of them. So this is just an example, and we would select one sample today and then try it on in the practice in the exercise 7. So before we get into the practice, I just want to show you what tool we'll be using. So we have this SRA toolkit that was developed and maintained by the NCBI, so part of the NIH program. So it's a suite of software that allows you to identify the specific sample information and then allow you to download it in a kind of high throughput fashion. So that's what we're gonna try today. So that's for exercise 7. So it's this, you can get to the web page through the same document we are using, which is, getting to the end of the file, or you can type in this new URL for today's content. So I just want to show you the QR code. So this is a QR code that is different from the one that we have available. So the PDF that you downloaded earlier are for last quarter. So if we use this QR code, that would be great for evaluation. So I can put it up later as well in case.
We need to download this page.
Oh, we don't need to download anything. So we're gonna use Hoffman too, and this is pre-installed. Yeah, so we'll just load the module like we did earlier. Okay, so I'll put up the website, the web page for today's content. Okay, so this is the material that we have been looking through in the last 2 days. So this is the file that has been sent around in the email. So the link to today's content will be available in at the end of the document. So that's application for day 3, and then you can click on that to get this new page. It's more kind of just to have a fresh page instead of you scroll up and down all the time. So for this part, we are trying to access public data. And then again, we're going to log on to Hoffman 2 and request the interactive node so that we can, download and then just let the memory on a separate node for that purposes. So you can click on this GSE, the geo accession number to get to the same page I've shown you earlier. So that's the sample we're trying to access today. And then there's another page trying to tell you more about the SRA toolkit in case you're curious about any other services they may provide. Yeah, maybe let's have a look at the website together. So for the individual sample, so Geo, they only host process data. So that means that, the recon table will be downloaded directly on the page. However, if you want to access raw data, you have to click on this SRA run selector. So that's where it's gonna get us to this page for the 1003 samples. So that's the page that I showed you earlier as well. So you can kind of look through this, this giant table with all the information that will help you guide through the samples. Okay, so we'll come back to the worksheet. So let's try to log on to Hoffman 2.
Do we need to move to the workshop folder?
Yeah, we can start going to the workshop folder, start a new directory so that the download the SRA files is going to be a specific subdirectory.
So when it says 1003 items found, what are these items?
So those are the 1003 samples. If you go back here.
Samples.
That means it can be 1,000 plants.
So somebody in the team collected
1,000 different samples. Yeah, that's right.
1003 sequences.
Actually, more than that, because not all of them worked, right?
Oh my God.
Yeah, it's a very comprehensive study. And then in the second part, I'll show you like what conditions they look at. It's very extensive. So they do the time series. So they sample throughout the day.
Bioinformatics information that are under public data.
Yeah, so that I think this is a great example to show just how how much data is out there and the wealth of information that is available at your fingertip.
So we are going to look at 1 out of 1,003.
We are just trying to download 1 out of 1,003 that is available for this data set. So far, what I figured out is for SRA, you have to download 1 by 1. I try to do, this kind of a parallel way, but I don't think it's working for me that well. So there are tools allow you to download multiple samples at a time, but you do have to check that the download was successful. So it's kind of quite a manual process. And I try to go through 200 samples in this one, and I think it took me a couple of days just make sure that I got all of them downloaded successfully. It's not that you can, automate the download process. It's because the download is it takes so long, because each of them are like 2-3 gigabyte, and the connection is not always stable. So, the Hoffman toolkit you cut off sometimes and you never know. And then, if you're using a lot of CPUs, they put you on hold and they will resume some later times. So those are issues on Hoffman too, might be want to kind of bear in mind. So it's not like I couldn't automate the download process, but rather is this process I have to double check the download was successful, that takes most time. If it's unsuccessful, you have to restart and then make sure you know which one is which one, right?
But you have to manually go through 1,000 of them to see.
I only dealt with 200 and that's about what I want to do.
How do you know that they're successful? It's going into the file and making sure.
Yeah, so there are checksums that you can make kind of double check. So we can have a look at the checksum or we download example sample here. Okay, so I hope all of us are on Hoffman too now. And let's try to load the SRA tools that are pre-installed.
What's option 2?
Oh, option 2, so you can copy the software, but it's already pre-installed. I think it's easier to do that.
Do you make a new directory for all these files?
Yeah, I can. Mm-hmm.
This SRA toolkit installation has not been configured.
Yeah.
It's not configured, it says.
What
This
is it?
SRA toolkit installation has not been configured.
On
Before
Hoffman
continuing,
too?
Please run BDD.
If you just copy the command and paste it, and then the default, and then it says.
Blue screen.
What?
Where it says you need to run this first, just copy that whole line.
Okay. That's why they just This is very interesting.
Yeah.
No, just copy pasting module load SRA tools,
Yeah.
and that's where you. No, the next, the prefetch. Prefetch. Yeah.
Yeah, it looks like you click on the.
Okay, so the replacement here. Run the next line, but it says. That's configured. Okay, so. You didn't make the directory that you wanted to. Oh, so the problem here is that page. On your code. So if you press up.
Oh, there you go.
Go back. Okay, so. Oh. Okay, let's register this again.
What is
So this is for sure. So here's an internet. So she said we don't have to mess with that one, just like that. If you press that. So you did the module load, that worked very well, right? Just took out that, we do this. It's this
Oh,
next bit. This is prefetch.
I see.
So that one instead of so if there's like
Yeah.
a hash
Try
after that,
prefetching.
it means it's like a comment rather than actually running it. So this is just a different option to do versus this. She said this is already on the Hoffman, so you don't need to do that. Okay, so don't, yeah. Okay, okay.
Right, yeah, so I saw the window. It's kind of funky. I've never seen that before. So hope you have, done with that. Just exit, click on X. I think that would be, that would do.
So it doesn't need to be configured.
So what did we do?
I think, once you've seen that page, it's configured.
And when it says what you need to do, that configure, copy that, paste that in, and we'll get like a blue screen, and then to come out that to trace that.
Right.
And then you've got to do the.
Prefetch again.
Thanks.
So I think it says, like, it says line. So it's this, you need that BDB, and then if you highlight up to interact and then run that. So you've highlighted it, which means it's copied, so now you should be able to right click on the next line. maybe go. And now you have to attach it. Yeah. Okay, so you highlight this B, D, B, all the way up to interactive. So this one. No, the next one up. If you highlighted it, that's obvious. And now it's on this one, you write it. Right. On this next one. Now press X. And then how do you refresh again? Is there my pick on that type of down? Yeah, mine's taking ages.
Yeah, it's a raw read. Yeah, that's why I was like, when you're dealing with a lot of samples, it's quite time consuming.
It's like a more general question. When we first log into Hoffman 2, I was able to see like the project and then my PI's name on there, like as some kind of directory. And then the workshop directory that I created, right? That we created here.
Right.
Is that like a normal thing to see, or are we gonna have to delete that folder, or does like other members from my lab have access to my workshop folder?
Oh, so when you log in, you're always in your home directory. That's
Okay, so
your
other people-
directory.
Okay, so other people in the lab don't know what folder.
I think others can see your folder, but they might not be able to do anything about it. So,
That's like when you set up a file as well, if you say it could be executable by all, just keep pulling over, like you can add that into editing files.
right, yeah, so they can at least see the file names, but they may not be able to view it.
Like what I don't want is like for other people to come to when they log into their that they see my folders all unorganized.
No, usually, so for example, I log in, I will see my home directory. I have to navigate to find your home directory to see what's in your home
It's not like there's
directory.
like a portal for the whole lab kind of thing, and they're like, Leo,
No.
this folder's all unorganized.
No, it's pretty hard to see that. So for the project folder, it's actually a folder in the project directory with your name, but not, you can see, of course, the other members in the lab.
Okay, I just don't want anyone to like see folders with bizarre names, like what is that? Why is it here?
Yeah, that's a fair question, but usually, yeah, it's not really a big concern.
Okay.
There are times that I have to go to other members' folder because they have a lot of the documents and softwares. So it's helpful to know, what they mean.
Yeah.
If you want to share resources across, the groups. So, kind of make legible folder names.
Yeah.
And so, the other will have an easier time navigating. Or, it's project-oriented names. For example, some of the trials, name, and then with different resources. For my case, there are germline data, tumor data, you better characterize them, and then put them on separate folders.
Yeah, that's helpful because I was like, well, do I have to delete my folder until I move it into the projects folder? Like I wasn't sure what to do with which is labeled workshop.
I see. Yeah, your home directory, I think it's it's only maintained by you.
Yeah, that
So I got, it was downloaded successfully,
Okay.
has 0 unresolved dependencies.
Right.
But then it takes a while to download to the working day. That
Mhm.
The next fast queue now.
Oh, right, the fast queue down. So that's a download step. And there's, like different parameters where you're downloading to. So this is creating a new folder called fastq. And it's gonna be gzips fastq file. So it's gonna be, compressed. And then we have a couple of other parameters, split 3, and then clip them. And of course, here is the files that you pass into the command. So that's a specific file that you're trying to download. Okay, so if download is ongoing, we can kind of maybe move on to the next step. So the idea of this exercise is that, okay, this is how you get the raw data from the web. You can feed them into the pipeline like we did in the last 2 days, just like, any other processing, any other samples. And then the idea is like, okay, now we will get to the recount table and we want to see what we can do with them. So let's go back here. So that's getting into the next step, which is trying to visualize RNA-seq data. So that kind of come back to the questions like now we have this table, and then table is usually oriented, organized in this way where we have genes on each row and then sample on each column. And then they're, showing different integers, hopefully, if not all 0. So now the question is, okay, we want to compare samples, to samples. How do we compare them in a fair way? So one thing that we know immediately is not all the samples have the same number of reads. So that we introduce a couple of issues is when we compare the numbers, let's say we have gene 1 for the 4 samples. Is it fair to say that sample 1 has more gene 1 expression, right? So that introduced us to the problem was like, can we scale the samples? Can we scale the gene read table somehow or normalize them so that I can compare between samples in a fair way? So one of the most, I guess a lot of you have heard is the CPM or TPM comp. So that's basically to normalize samples so you have basically the same number to begin with, which is a total number of reads. And then you scale it so that you are actually reporting the relative reads for individual genes. Imagine that each sample has 1 million reads. So that's another measure would be this kind of TPM, which is transcript per million. So it's a related measure, but they are pretty much the standard in the literature nowadays. And then for, if we want to do direct data visualization, we have to think about a couple of issues, for example, between gene differences and between sample variations. So between genes, of course, some genes are, more expressed than the others. So within the same sample, I think that's fairly straightforward to say that, oh, gene 1 is expressed at a higher level in sample 1 than gene 3, for example, right? But if you want to compare between samples, I think we'll have to normalize it quite rigorously. So there are different methods out there. And maybe some of you have tried the, couple of different tools. So one of the tools that is widely used is the RNA-seq. So what they do is this kind of trying to find out the size factor. So the idea of normalization is we want to make sure that each sample have been scaled so that as if they have the same read to begin with. So there are a couple of concepts, which is, oh, we can scale it to the median. So that's one way. And then another thinking is, oh, we can, scale it to the mean if we're only counting for genes that have, non-trivial expression levels. I hope that makes sense. So another kind of key concept that's widely used is this geometric means, where they are kind of trying to find out, is there kind of a meaningful way to represent the average, basically? People try median, people try mean, people try geometric means. So it's kind of different combination, but depending on what you want to really take care. So usually people are happy with the D-seq2 kind of normalization. Basically, you take the median, just take all the genes that we have. You can filter it beforehand. For example, if some genes are having 0 count, we throw them out, something like that. And then you take the median of the remaining genes, and then we normalize according to the median. And one thing that I like quite a lot is this TMM, which is trimmed mean M value normalization. So what they do slightly differently is they actually only care about genes that are not trimmed based on the raw read. So that means we only care about genes have meaningful readout because a single experiment can generate this recon table for up to 60,000 different gene regions. So 20,000 of them are known genes, they're annotated, but there are the pseudogenes regions. For any experiment, usually you get around 10,000 genes that has meaningful expressions. So that means that instead of, trying to normalize based on all the genes, we only care about this around like 10,000 genes that has actually real value, that's non-zero values or non-trivial values. And then you're trying to normalize based on the sum and the mean among the genes that are not trimmed. So in the end, we will get to this kind of Z readout. It's more like, a Z score where it has been, the gene expression level has been scaled so that we can compare between samples. So I think this is good table to look at occasionally if you want to compare different software outcomes. So I would say this is quite a standard practice using D-seq2 normalization, but I do think Agile, TMM using Agile has a lot of other advantages. If you want to say that, oh, what if some genes are longer than the others? It has a higher chances of, mRNA species mapped to it. So TMM also counts for that. So D-seq2 might not really take care of that aspect. So the data that we've downloaded are from the soybean RNA-seq experiment. So what they're trying to do is looking at the plant, the leaves, and looking at gene expression at different time of the day. So they sampled 6 times within a day. So every 4 hours, they have a sample. And then they have, different time point. And then for each time point, there are different number of samples in bracket. And then they also sample the different treatments across 7 days. So especially for day 5, they've also sampled leaves at different time points throughout the day. So what we downloaded was for the day 0 data. And we want to actually make sure that we can somehow compare what's going on in the baseline condition. And then another thing is they have different condition, lighting conditions. So one short day, short exposure to sunlight, another condition is long exposure. And we're interested to find out whether there is going to be a difference in gene expression. And then they have 2 samples per condition so that we add up to like 4 for each time point. So here is this kind of an example in terms of how we visualize the data. So on the left, we're looking at the day 0 data. So it is cultured under a 25 Celsius condition. And what we have is these 2 conditions, long day versus short day exposure to sunlight. And we have a readout at each time point out throughout the day. And then we have genes on each row. And then we can cluster them based on their kind of group relationships. And you can see the blocks already. Basically, we have cluster of genes expressed early in the day, or they're kind of, highly expressed in the middle of the day or late in the day where, they don't change at all. And you can start to see that in different conditions, they have slightly different patterns. For example, the chunk here, it seems only to be highly expressed in short day exposure to sunlight and not so much expressed in the long day exposure. And then some genes in the early, peaking genes seems to be lost in its expression if it's short day exposure. And then we can mark individual genes that we are interested in. So those are a few genes involved in flowering and circadian rhythm. And then we can also compare, of course, across different days due to treatment effect. So that's about it. And we want to try the soybean data. So what we have for you is actually all the samples that we use for the heat map generation. So it's already being processed. So all you need to do is download it from the Google Drive. So let's go back to the web page. So for exercise 8, we'll try to cluster and do heat map on the soybean data. So we are gonna do this in R, and hopefully you can have R Studio installed on your laptop. So we're not using the Hoffman anymore.
So what, do you have to transfer files from Hoffman to your computer?
No, not for this exercise. So this exercise, everything is gonna be on Google Drive. So just download it from the Google Drive, and then we load it in the laptop in our studio.
This is basically right after all of this stuff.
Yeah, so for example, like yesterday, we have gone to the recon table. You would have downloaded the spreadsheet to your laptop, and now we do everything in our studio. We don't need Hoffman.
Your local.
Yeah, yeah, that's right. Okay, so let's give it a try.
Very totally done with Hoffman, or-
So if you know you are still downloading, you can put it aside, but you do need to kind of open the Art Studio on your laptop now. So that's gonna be the main focus for the remaining of the workshop.
Question about yesterday then, the pipeline,
Mm-hmm.
it was supposed to generate a count table?
Yes.
Because I feel like the script I had, it only took it to alignment. It didn't have like the downstream.
The pipeline? There's a separate script. Did you do feature counts? Yeah, we can have a look together. So that's the pipeline here, right? So we open it up. So step 0, prepare your sample. Step 1, set up working directory. Step 2, that's quality control and gene alignment. And then there's a step 3. Have you gone through the feature count script?
I don't know.
Yeah, so after alignment,
Oh, because I guess I submitted the batch and it didn't seem to.
and then you went home, I guess.
No, I stayed, but I guess like I don't think it actually it didn't run. I have the files, but I didn't generate
So do you get the BAM files out?
no.
So the BAM files are the output from the alignment script. So after you're done with all the samples, you can run this feature count script, which would tally the per gene count for the couple samples you have.
Okay.
So this is gonna be a separate batch job,
Okay.
right? So if you carry on with step 3, we should actually get to the point where we actually have the count matrices out.
That makes more sense.
Okay. Yeah, so today is more kind of, okay, now what do we do with the count matrices, right? So we'll try the TMM normalization. So we have a soybean data, and then you can download it and then see how many samples there are.
One issue, just installing packages, the package.
Right. Yeah, so for our studio, if you have not installed all of the packages, we'll have to do that first. Mhm. I think it's downloading now, right?
Well, it didn't finish, but then all of these errors.
No, oh, is that error? I saw this one.
But I tried to do this afterwards, and then it says that there's no package called data table, so.
Yeah, I don't think the download was successful.
Yeah, but this keeps popping up.
Mhm.
Like, do I even select yes to this one? Try.
Yeah.
And
So yeah, it's like this thing, and then I don't want to like, because I'm like doing exercising now. So I think like, yeah, now you're working in R for this bit.
You're.
Yeah, I think so. We're gonna download it.
Yeah, I think something went wrong.
From the Google Drive. Yeah, yeah.
Yeah, you have to troubleshoot a little bit, like why it didn't install
Yes, it's a-
successfully.
it's a normal.
Yeah, it was quite straightforward for me when I was doing that. And I'm using the same version actually.
Yeah.
So maybe there is some GCC issues with your laptop.
For GCC.
So so one of the errors says it needs C++.
Yeah.
So you'll have to actually activate it or try to find on your local laptop. Mhm. Right, it says, that, yeah, you need the C++.
So I need to download what else that is downloaded.
Mhm. So now you're installing the source package. Yeah, which is.
Like the initial install.
Really? I don't install, have a try the Edge R.
Yeah, I think it's the library. Do you have Xcode?
X what?
Xcode? Yeah, I think it's the laptop configuration. So sometimes like for R, we're trying to install things need C++, then you need to GCC package active.
Okay.
So Xcode usually take care of that. It's more kind of you for like program developer.
Okay.
Yeah,
This is just
you-
automatically.
I think like when I have that installed, it take care of the GCC background.
Okay.
Yeah, just try that.
Okay.
Yeah, it happens all the time. So which R are you using?
I think it's because you need to install the file manager, not with the install file.
What's it called?
File manager.
Yeah, let's put it up here.
Someone to name it.
Right, so you can Google that. So you can use the Bio Manager to install Azure. I guess most of us have experience working with RStudio, right? I was like entirely new to it. So trying to get packages and then run script.
What does it mean? What's the dimension of the? The two-dimensional. Now we're just trying to give normalization stuff.
So I guess we have downloaded the data set and I hope you're fine with the packages. So we get to the download folder in R so that we'll be able to locate all the files that we have just downloaded. And we want to load the data into the environment. So we're using fread, which is part of the data table package. And then for TMM normalization, we're gonna use HR. So hope the installation was smooth.
Where's HR?
So HR, we were, if you have downloaded, so it's gonna be just loading. And if you have not, you wanna install it with the bulk manager.
If it doesn't give us an error, is it?
Yeah, so if you have already installed, loading will be smooth. So when you call this, it should not give you error messages saying that, oh, we cannot find that package. So that's pretty much the tool that we're using for normalization. So the other tools are pretty much using for manipulating matrices. So dplyr is what I usually use. And then for matrix stats are for calculating mean, variance, standard deviation, etc. And then the last two are for plotting, which we're going to be using to generate the heat map. So if we come down here, hope we can load the data successfully. And the normalization step, basically, we want to create a sample object. So that's kind of involving creating, loading the matrix data into this object called matrix of data. And we want to create this DG list object. So this is a well-defined object for Azure. And it's very similar, you were using D-SIG2. So you want to specify where the count matrices is and whether there is a group relationship. For example, we're interested in the differences between different time points. So we give that as the information to the group annotation. And then we want to filter a little bit. So the filtering step is one line of code, but when you're dealing with your own data, you will have to play with it quite a lot. So can anybody tell you what this means? So we are trying to find out the CPM of our DG list object that is above 1 for at least 4 samples. So that means that, we're trying to keep the indices, which is genes, that have at least 1 CPM in at least 4 samples among the 25 samples 24 samples that we have here.
What's CPM?
So CPM is count per million. So that's the measure we have introduced earlier. So this is a very basic filtering. Basically, you want to make sure that we only care about gene that has meaningful reads. So count per million above 1 means at least 1 out of a million reads. Otherwise, it's going to be lowly expressed and we're going to just filter them out. And then we want to make sure that, it is at least, above 1 for at least 4 samples, because not all samples going to have high expression of this gene, right? For at least some of them, they have high expression, so I can capture some variation as well. So that's what I usually do. So when you're dealing with your samples or your data set, you want to play with maybe the baseline CPM cutoff, or you want to change, the proportion of samples having at least some read for this for the gene. And then we, count how many genes are we keeping. And then, of course, we subset the DG list object so that we are only keeping the genes with, decent expression. And then we check the dimension. The dimension should give us, the number of genes that we're keeping and how many samples we have. And then for DGTMM normalization, we will have to go through 2 steps. The first step is to estimate dispersion. So dispersion is kind of another measure for variance. So we're basically feeding a distribution to the data so that we can calculate the normalization parameters more accurately. So this should take some time to go through because it's basically doing, manipulating this giant matrices, trying to estimate the variance dispersion. Then there are individual commands that you might want to check what they're doing. So we have calculating normalization factors for this object D. So D is a complex object. So when you're calling this, it's actually adding a normalization factor to this list object. And then for the remaining lines, it would pretty much estimating common dispersion and then tag-wise dispersion. So as you go through, run through this couple of commands, it will give you outputs to the command line, and you can start to see, how does your data behave, and then you can adjust accordingly. So after the dispersion estimation was finished, we performed the normalization step. It's pretty much the very last step of the multi-step process we have talked about earlier. So here, that's the normalized data. Basically, is getting the pseudo counts from day 1 object, which is the ones that we have done earlier. And you want to write it out as a data frame so that we can manipulate better in R. And you want to add a gene ID to the matrices, so you know which row corresponding to which gene, so you can check manually if you need. Of course, at the end, you can write out this normalized data to a CSV file, which is separated by commas. or you can write out as a plain text separated by tab or space. So that's about the standard readout, kind of write out document in R. So after normalization, we can start to do visualizations. So maybe we can start trying the normalization step.
Do we need to add?
No, so once it's installed, it should actually help already.
You have to reboot.
Yeah. Yeah, just restart R. So just like that one. Oh, Oh, I see. Yeah, just try the Mac.
Oh, did you? Oh, you did. Oh, yeah, there you go. Yeah, must be great. It was like looking for. Two things. It was a Halo G dot. And then also it was like a change. Which, not that. For like, is it a while? And then they just really like the. Yeah. I'm looking to it. Wait, I understand. Okay, so this is just in. Yeah, we can get the paste function
detail this function.
Right.
Because it couldn't find it.
Did you create it? You have to create it first. Yeah, just a quick note for setting the working directory. So we are in the R Studio. You can click on session, set working directory and choose directory. And then you can pick anything you want from the list. So that's just a quick note for some of us. And for creating.
Oh, you have to load the edge on. Try again.
Yeah,
That's
for
why I don't
Edge.
have the function.
Yeah, you have to load the package first. So for Azure, you have to go back. So we installed using this function instead, right? Because Azure is a very extensive package. It has a lot of dependencies. So a lot of kind of very complex packages now maintained with the bulk manager instead. So CRAN, support all the basic functions. But when it comes to, those kind of rocket, science packages, we usually go with the bulk manager to take care of the dependencies issues. Okay, so I hope some of you have gone through the normalization, and we can, even try some of the visualization code. So the idea here is we start with this giant matrix where we have how many rows? We have 52,000 genes, and we have 24 samples that is on the columns, and we want to process it first in R. So depending on how fast you've gone, so here are the code for running the normalization step. So just make sure that you have all the packages loaded successfully so that you can run the individual steps. And then later on, we can try to subset genes that we want to visualize. So what we are feeding into is the flower genes. So we are selecting the 72 flower genes, and if they are in our data sets, they will be subsetted. So that's what this first chunk is doing. And then for the remaining part, we want to somehow, aggregate the data, because we have 2 samples per time point per condition. So what we are doing here is pretty much group them together and then take the mean. So this is kind of this big, chunk of code is doing. So we want to put them together, and then we take the average for the 2 samples. So we do that for long day and short day condition. So it's quite repetitive. So it's kind of the same, just with different samples. So after we have done that, we calculate the z-score for the expression, so that, the relative expression is going to be on the same scale, so that when you are doing the heat map, it's not going to be like one gene is super bright, another gene is super dim, and you cannot really see the variation. So one way to go around that is we take, we find out the mean and the variance of each gene variation across all the time point. And then we calculate the z-score, which is a standard way to scale the gene expressions. So we aggregate all the data into this giant data frame, and then we can write it out or proceed to heat map generation. So for heat map generation, there are a couple of standard ways of doing that. So one thing we have to do is go through this hierarchical clustering algorithm. So it's quite straightforward in R. And many of us are actually going to ask, like, what are we actually doing here? So it's kind of one way to cut through by comparing how similar different samples are and aggregate them in a tree structure. And then by deciding how, how far along you want to cut the tree, you can group them into different clusters. And then we want to specify different colors for the clusters. And then of course, we want to choose a palette for the visualization. So here I'm using the red-gray palette. So you can choose any other, like, fire or red against blue. That's quite standard for publications. And then of course, the last step is to plot a heat map. So the heat map will be kind of, a giant block with many tiles, and then individual tiles represent just how high or, how big the expression is compared to other time point. And this is how they would look like. So when you output the heat map, they would not be so clean looking. So this you have to add some final touch. So what I did is to load the picture into Illustrator, add on, the time information, and then try to group them. And then I can highlight genes of interest within the heat map. So hopefully can kind of get an idea about, the process of how we generate figures and then prepare them for publication. So that's for exercise 8. So let's give it a try. I think like some of us have already gone through this part. So if you are happy with it, you can kind of wait for a bit, or you can proceed to exercise 9, which is trying to visualize a time series in mouse kidney. So that will be generating some sort of box plot like this, where we have normalized mRNA expressions for selected genes at each time point for different categories. So here we are looking at male and female time series. And then what we are trying to introduce in exercise 9 is one way to automate this process. So with one script, you can generate this figure for any genes that is included in your data set. So this data set, I think I have around like 13,000 genes. So it's one way, it's like, okay, I don't know what gene I want to look at in the future, but with this same code, I can generate any plots in basically 2 seconds. So what we want to do is to have a look at this script, which is a scripting R. It's like running the bash script we did in Hoffman 2. You can run this R script on your laptop command line and just give it a try.
Got an error before getting to the z-scores.
Okay. Okay, this step.
Hierarchy clustering. Yeah. Oh, this? Yeah. Yeah, I see. So just. Thank you. Yeah, so now it's very, we have changed. Yeah, for both of them. So. And so we actually push it down in the cup. What do you got? Oh, I see. So maybe you stay in it.
Yeah, I think. Yeah.
Yeah, so every time I start a session, loads of technologies. First, the first step, yeah. I convinced, was like the copy, it just gave me another. Like, bio manager was a wow. Yeah, well, I did everything, this whole.
Yeah, you can do wrong, I think. Yeah. I have an heat map, but it doesn't look like things.
Like the labels are missing.
Like the general where the reds and grays are.
Yeah, so you have to touch it up. I
Like,
did it
it
in
doesn't look
Illustrator.
like, you know, I don't have all that green.
Yeah.
It's smaller. I have like 2 types of green like this.
Oh, I see.
It's-
Yeah, so the color is somehow random. You have to figure it
But
out.
then also this, like the gray,
Mhm.
red, like I don't seem to have the same patterns.
Well, it's the same data. Yeah, one day. So this is that. It matches.
So it's like, so it's flipped? No, because I'm,
No, it's the shift is chunk, it's that chunk. It's a different plot. Right.
yeah,
It's you actually should be that.
Yeah.
Yeah, and this is that.
so I did get, this error warning message.
Yeah, that's one.
This font is different. I guess we have to delete something as well. I think it's, no, I didn't do it.
Yeah, it looks like it's more than that. The classroom should be the same. It's, I run it a couple times.
It's like it shifted it around. Like what? This is, okay, 4 colors.
That's chunk, yeah, that chunk is that chunk.
This is like. So in Illustrator, you do like this separation as well for the lines.
Oh, I just added it. So it's clear. Yeah, but I guess, you know, like, get a sense, how we get maybe that look back. Yeah, well, that's helpful.
Yeah.
Yeah, HR even. It's small projects. I'm setting it up to do it. Yes. Yeah, have you done it with
Okay, great. Okay, sounds good. So now we can proceed to vote. So now we vote them. Let's try this. We have two votes.
Yeah, that's me. Yeah, I got the job. I never want to do. I know I had it. That's the place where-
Yeah, I know. Yeah, try the pipeline, hopefully you can get this.
Yeah. And
Oh, yeah. So you found like some.
Yeah. Yeah, so I'm gonna go through.
Yeah, they have to finally get them. Yeah, try to find the information files now, and you can do the next steps. So once you have them, you can just so run for it.
And that's just using.
And then you can try to visualize the different times.
Yeah, I think it'll be good. Hopefully. So you can find the data. But thank you so much.
No problem.
I'll send it to you in a second. I got to go through it. Thank you so much.
But the labels, the timing labels, can you specify something else in the heat map?
No, so the heat map, that's, all we can do. They are kind of clunky, I know, and the trees, you definitely don't need them in visualization. So what I did is I loaded the image into Illustrator or PowerPoint, can do that too. You crop out the part that you don't need, and then you add the little labels like time point, groups, and then highlight the genes of interest. And then maybe you want to add additional lines, kind of a partition so that it's easier to see the two conditions. So this is kind of, more the aesthetics so that it will make it easier for readers to make out the message.
Have you added significance here? Or like the stars?
Oh, the stars is more kind of highlighting the genes that I want to showcase. No, there's no stats done here. Yeah, I can use like, cross, any other symbol.
Oh, oh, you can do roles with FDR. I think we should throw these on that. It's the cute. To make sure you know. So the types of movement we can repeat. Yeah.
Oh, that's yours?
Yeah, not much. So when she thought it was slightly different.
Right, so what is that?
And the matching with it.
Yeah,
some other colleagues didn't do different, you know. Really, that's so strange. Really strange.
So she copy paste.
Yes, I thought so.
But she doesn't.
Oh, that is scary.
That's scary, right? Like if you just, installations.
Yeah, because the hierarchical cost, you're gonna get something, you know, it's not stochastic, and you could go and you see, then I'll get. No, I mean, it's not stochastic.
Yeah, if it was,
It should be, yeah, it should be.
Yeah, I'm just trying to even look at like the tree thing on the side and how it compares. Not even looks at it.
so in my tree is then.
That's what, yeah. I was just thinking.
Oh,
Yeah, they're very different. The colors are different, it's sorting different colors.
You have like a really small blue, and I don't seem to have one that's size.
What's the secret search?
The green looks to be the same.
Same size, because it's like even size, right? You've got that one at the bottom is really big. I don't seem to have one.
And it's not a matter of my size, but no, it's
It's a different structure.
Do you get it to space?
I got it from the Google. Yeah, it should be the same.
and it's the same file name.
Yeah.
That's a mystery.
Your grays and the stuff are also different, which is not surprising.
Yeah, I feel like you're applying with different names. Yeah.
Interesting.
Do you have your tree? That you could, we can look at your original tree. That
Yeah, I found the wrong one.
It's like.
So many pictures.
I think like the problem for you was like, it seemed like, because you loaded it and you didn't get an error, whereas like mine, right, for some reason our studio, I always get errors with like right store, it's always like, so that's why I managed to back it up thoroughly.
Proactive, mine was sitting on it.
Okay, so yeah, you,
Yeah, so that's the raw figure I put into Illustrator.
At least with the color distribution is similar to mine. And
I got completely different.
I really suspect I have a different data set.
Like, especially because I had to delete an NA row.
Yeah, I wonder what that is.
this is identical to yours. So following.
Uh-huh.
So
What can I say? Happy
You-
people.
Somehow you clicked into a different universe.
Yeah, it sounds like.
But there were so many. Like I remember that, what was it called? SRA. This one selector, if she just like shows in a different.
No, but
Not
this
even that,
quad
it's the
was
difference.
using the same recount table, right?
is-
The matrix,
Oh, yeah.
the same input. It's a very interesting scenario, no?
Oh, yeah, so we saw this earlier. Yeah, I tried to do that. No, it's the library.
So now we're trying to analysis.
No, it didn't, yeah, it just started, we did the last thing. Yeah, it just did that. So you can continue.
So for the last part, or
Mhm.
the command line.
Yeah.
To do the.
Okay, yeah, have a good time.
Are we doing it yourself now?
Yeah, so if you're done with 8, yeah, proceed to 9.
Yeah, so you download it to your office, right? Yeah. And then go to your download, go to where the file is. Where the first we download. So here's two. Yeah, I don't think you downloaded it to the download folder. Maybe you do. You go back to and you're in core. Okay, so you download to the core folder. Go to your file. Here. Yeah, this is another way to do it is to go back to file and then select the download option.
Got an MTX. I don't see those pictures.
Okay, so now you were downloading.
No, I think MTX is this one that you just made. So you should, it should be in your history.
Yeah, so now it should be your download folder. Yeah, so file transfer. So go to your download folder.
So it's fine if you go, like, if I go to environment. Or-
Okay, so now here we go. Now just open it. Oh, okay, thank you.
Then you can scroll down to M and look at MTX.
Yeah, so it's like a number. So I've got here 1 to 79. Well, I have a like the main line is not the best to work on because I managed to. Scroll through. So the RS-script plot the trace command R automatically. So this is supposed to make this work. It knows where it is access. Yeah, so we can open this file. See the two files. So using the execution, the RS script for. Oh, that's the full script, right? And it's reading two files. One is the one with the normalized speed, and another one is the meeting and. So that's the two files where they have the download them in the same folder. Yeah. So when you run the R script, basically, it's kind of running the script in the for the specific gene. So I did the specific so then it provides us with not in the same sample. I don't think it ran. So you have to do this in instead of you get the terminal here. I did that too. This is maybe I have to go to what I'm on Hoffman right now. Oh, it's local. It's a local terminal. Yeah, you don't need. I don't need a Hoffman. Okay, so. Sure. And find this. Yeah, it's done. And it makes that in one. Quite magical. Right, so I'm just thinking, I'm plotting too many. I'm gonna make a script that, churn out the figure I want. That's good to know, so you can't do that. So for your own data set, you can just use the script as a template. Yeah. But you wrote your own script. Yeah, but that's not trivial. None of it is trivial.
Yeah, but when you're working with it every day, then it becomes one go. Like you wanna do it in one go.
So you can run R. The scripts on terminal, like you know, I thought you have to run it. Why couldn't I run it on R script? Command. Why couldn't I have done it on R Studio? You did that in
Oh, it's a different console. Our script, it's for bash, not for R. Doesn't make sense. So it's like running R in a bash environment with this script. Put that input.
Could you have written your script in R and then run it in R?
Well, the thing is, you can just run this script. You can just run this whole script. That's it.
So I'll go to highlight everything.
Yeah, but you will have to specify the argument. Remember?
Let's see. But these are the arguments.
Yeah, so if you're trying to do it within our studio, you just change this to the G names. Just that. Right.
Where did you do that? In the terminal? Is that
The terminal,
thing you had?
yeah, the terminal was just the input, and it's just the input.
So GSTA2 was the name of your G.
G, yeah.
So that's what you wanted to plug, so you have to then change that entire studio.
Yeah, if you want to run it in our studio, that's something you have to input. Right? If it's running it on terminal, that's what I fit it in. So, you know, that's like run this script and then you specify which script you run and the argument, input it into the script. Yeah, it's more kind of just trying to do the same thing in one. For example, like if I'm doing this, I don't need to open our studio. Oh, yeah, so have you loaded the package?
I managed to make it work.
Go back to screen. Good, I didn't know where to do this.
This is for she's written.
Yeah. Yeah, so then you really need that G.
Sure, it's that particular G.
Even if you do, you can copy paste it.
We run that.
Giving you don't need that arrow.
Once you run RS, you can change the G name inside files. When you go to be able to run different G events, you just have to change the name in the command line. In the command, you just change it, put it in the G there.
Yeah, it would, and then it give you different data.
It gives you different. It's a large for that particular G.
We'll double check that everything
I want
runs.
to try different.
Yeah, let me try because we only tried that, right? It's the.
Because this one.
Yeah, you can finish the remaining from this one.
That's nice. Now it's. It was like.
Yeah.
Okay, yeah, so
Find
that's.
the G names, but I'm not there yet.
So now you can proceed to clustering.
Yes.
Then you know. Oh, yeah, so you have to load the packages So go back to the web page. Yeah. Yes, so you can family level. Try to know what's happening. There's a lot of it has the same kind of a structure and the same. You remember at the beginning, yes, something was for the same Hoffman, that was supposed to download. Yes, sorry. Yeah. So this work. I got a fake of
Yeah, mine said it downloaded, but I don't.
We are trying to get that way the long solution. It's just fast, you don't click the error code to read.
Oh. Mine said rejected some reads. It's a filter.
And then we load it again. So it's loaded successfully. So we'll continue the heat map part. Let's try to rerun the whole thing.
Well, I think I got
Yeah. So now error code for your folder.
but I just did it right now.
Yeah, right out.
And then once that is finished, if the fast cube dumpling, and then it was helping. It looks like I got the page to learn, or I got an error code free. So I have somebody finish again. Let's see.
I think it's a download folder. Can you open the folder? Yeah, I think that's, yeah, that's the, okay, yeah, so that's the output that we just, plot. Yeah.
Did it work?
Yeah, it's working.
I'm just so close.
Yeah, well, we like now you know how to, start from the beginning and then try to get packages, load the packages, and then run the individual steps. So it's always good, you can revisit to see what each line is doing. And then if you're, using the code in the future, you would actually have a better handle. It's like, oh, you think something is, going wrong, I know how to fix it, I know how to adjust. Right? Because when you're doing data analysis, there's so many parameters you have to change according to your own data set. Yeah, you have to fine tune a lot. My pleasure,
So anytime I
Yeah, every time you open a new session, for example, I start my laptop and I open our studio.
always start with loading packages. Yeah, and then try to read files. Yeah, the very first thing. And I always give it as the first thing to do.
I have to go, I was trying to go back and finish off the,
Oh, yeah.
yeah, pipeline, and I got, Sarah in the job. I guess like it was like what I specified, I guess. So I tried, this is the first receive files, and then.
We have to look into this. The battery.
Oh, yeah. Change this, I didn't change anything else. So then, this is modules exporting from.
Yeah, that's hashed out, so.
Oh, yeah, we're not doing that.
Yeah, what is that?
Oh, and then so it's going star alignments.
Yeah. So you have this folder, you can come.
Not in this, actually.
Yeah, that's for yesterday. So one thing you can do is to create a comment folder.
Like it was actually in. It looked like it was supposed to be.
Yeah, I think what is going on for this folder is you have. Oh, yeah, I think I have a wrong idea what's going on. So the script that I have for the pipeline, it asks to create a management folder.
Yeah,
Yeah, I think skip that.
I made it, but I think it's in my like workshop folder, and not necessarily in a. One for the day. That's when I got to this. And I moved things into.
Right, so I would recommend to try the pipeline with all the folders created. Yeah,
So like making you. Okay.
for example, like you do everything in your test folder, but just make sure that you. Yeah, that's the thing you're using for feature camp, right?
So can I copy each the down?
Into tests, yeah, into tests, and then
How can I copy it from this folder into text? That's something that I always struggle with. I just end up using. I mean, it's like that. I should see folders, but it's.
oh, you can do it, yeah, just need to come online, you can go all in one step. And then just CP. Send it to the slash. Then you copy. Yeah, where is it? Be a different folder. Is in your process, yeah.
It's 1 hour.
Then which we have. The process.
for that now.
Now you create a folder. Yeah, now it's ready to go.
Right, so now I So we made this, which we were supposed to make this now, but we're not doing it.
Yeah, it's just the example. Right, you have one. You have something there. Invalid.
Sure, that's right. Airlines and.
Yeah.
The last two copies I work. Okay. And probably because of the issue with folders.
Yeah, maybe we could find, oh, yeah, the post QC file was somewhere else.
Yeah, okay. So that's probably
Right. Yeah, so if you move to the post QC and then try the alignment.
Good question. I'm having trouble getting this to run through command prompt, and I was trying to find what to change. The actual bars. I see it's like the email, pretty sure. What would you change it to? Just
I wouldn't change it. Yeah.
just
So that we would able fine, I just submit.
I know.
Oh.
I'm doing it now and I did it at home too, but it didn't matter. Apparently, I submitted a job, it started and it was 12:30 when I said it submitted, I got an email. Closed my laptop, came here, and I never got a response back about it being done.
Is it still running? You see? It's my action.
Oh, he's got what I just got something complete, but then. Maybe that's the one. And then I started it again. So I always, every time I start doing something, if you stop, I get two completely one sort of beat, and one I started and it completely first, and then it started this next.
That's probably means that I do. Yeah, I think if it does that, it basically, like, if they come through at the same time, it's like the job started, it's stopped again, so it's probably it. And that's what I think I got as well.
So where was the error? How did you stop it?
So if you look in the job log that was probably made, it will be made in the folder wherever you are.
My job, my job.
It should be under like whatever folder you submitted the bash file from, the sh file.
And that part.
Yeah, and then the media, yeah, the rest of the command is
So like, and I'm going back to like exercise. Depressing. So for me, what I did was.
yeah, so just replace.
What I found on my document, for example, was.
And then the gene names.
in this, I made other folders for this next step, but yeah, she would have it in this next step. So I like folder you yeah, and so I think what I did is I did make that, and then I have files, but then maybe I submitted. Yeah, it was like changed, say if I show you, like in my, if I changed to my past.
So you might have to define either path.
They are there. Yeah. And it's by, and it's that linked file, so it's not.
Yeah, so apparently something that was going on right now, you cannot find our script.
Yeah.
As a function or as a software in a prompt.
Yeah.
And so with the website, we can supply them.
I signed off, but I didn't,
Right?
yeah, so then I submitted my, it was the.
So I need to.
Oh, it's actually,
Where that.
yes.
If you have R, if you have R school, then
Maybe the.
I have to make sure that you.
Yeah, I think that sometimes, I mean, you can specify that as a batch file, I guess like.
The worst of worst things that you obviously do is certain words,
And then you don't change it. So if I, I'm gonna look at this.
Oh, that's
Yeah, that will take care of it.
Got it.
Then I guess what it.
So instead of arguing.
Yeah, that's. Yeah, I can just, time whatever change that you want to plot.
Do you?
Yeah. How are you? I haven't talked to you for a while.
I know. I have to go into the corner.
So I need to get that to work. Oh, cool.
I think I'll go.
Yeah.
I thought
This one's right now, I think there's somebody needed that.
Oh, great, yeah, that's pretty much all I'm hearing after techniques.
Come on,
So I think she says here, like, so for this, she says, give it a.
Yeah.
So it's like,
Yeah.
and this is when you submit the job.
Well,
So you queue stuff,
Right,
with
yeah.
that is extra. I don't specify anything.
Right.
Yeah.
You're ready to do that.
Yeah.
No, you do.
Oh, which don't change this.
No. Like, and you say like you want counts, then it would.
Yeah, so that's what she
translate it.
And did you
I
just?
think it's like. It's that's the first time we've done that as well, because the last time. Or maybe that was what we were doing last time, it was specified it. as not.
Yeah, so
Maybe it wasn't anything. I thought it was looking at the file as old now.
that's what it takes a second. Like 2 seconds. So they exactly. Yeah, just change the name. and
So I think my problem here is where it's the picture, but it looks like a brand of out file. It's not to be safe.
Yeah, I think you're elaborating some sort of consideration. Yeah, that's what I think that. So you can just go around, that's a good start to be able to.
Wasn't super, yeah, organized, but yeah. Mine's not.
Not the laptop, upgrade the R Studio.
Yeah.
Sometimes people previous stats.
I just got.
Oh, yeah, but yeah, maybe. So I'll recommend. Everything. It looks like your.
I think that's the problem. In the script, your.
And then you're the one.
We don't actually do processing.
Yeah. I have to set up.
No, I think this is the 2 stages. What she wanted. I think.
you know,
The first thing we ran was this, the sum so okay, sum is that. And that was like quality control again. This was I got around to true. I think you were doing, I remember doing this yesterday. You know that we finished this and then that's why I feel like I'm
Okay, so just-
really to be aligned with it. She was like, oh, you
Or at least because
yeah.
I,
Mainly because I was like, oh, we're not going back to it. I thought we were going back to this thing and then finish it. Yeah. That would have been nice. But I understand.
I think everything else is fine.
Mouse liver, mouse kidney, soy.
But it's
I mean, I understand why. It's like now, down the line.
Do you know what we're doing?
Yeah. Well, I'm still like.
I see, yeah, so let's give it a try. So those things are gonna be too much, that I love us to, so hopefully one that we should.
But yeah, so I think for this, I need to.
Yeah, basically,
I think
more is more for you.
what we found, just looking back.
Okay, so the whole.
No, so I never, I just tried to.
Like, one of the one is funny.
No,
Yeah, I'll report the whole sort of way.
Yeah.
And they're both points, so it's okay. It looks really bad. We look in the past, we had like 8.
but I don't know what's gonna run because.
I'm
What we.
yeah, this one's perfect. I'm not.
But the bounce. Well, they could do the bounce, that's not that, but I think I said. You generate, it started to run into the line -0 because.
Got it.
And you'll look right, so here we're like, maybe you do LMs.
Yeah.
And then, that's the thing. I really good stuff.
But- I was reading through it. I was reading all of this.
Yeah, it's pretty much the same thing.
Yeah.
So maybe go back and then just check. the content. Oh, okay.
That's it.
Yeah, so do LS.
I see, so then here too.
Yeah, well. So,
So what you wanna do actually.
So I get always a really good way to get it. The single song has, you know,
The other way to do it.
Yeah.
If you do.
yeah, it's a good foundation, but I still have to work on
This will get the size of the files. So now we know we had this came from.
Oh, you can check anytime. Yes, I'm not teaching that here, but I think I what I'm function work.
I have a. I have a great and I have to. Sorry. No, it's just, oh, you've got a second. So.
Yeah, one second.
Sorry. This is very sorry, I'm just trying to reload this part and then I see that I want to change like some sort of lines, like I'm not changing the entry part to like 15 or 16. And I just like, you know, once I found here, it's a copy paste and then I know how to go back to changes. Yeah. I'll try to like run this.
You can control C to cancel.
Control C.
Yeah, so you cancel that. So you can edit this if you have a text editor, maybe something.
Text. This one? Oh gosh.
New document.
Oh, new document. Okay.
And then you paste, you copy that.
Copy.
Yeah. And then paste that in, and they can change.
Okay. So let's see. Oh, what's the question? Oh, range is going to be 60. So the character is 60, and this is 15. Yeah, just do 2. to.
So the key of the PCR
And then see and paste it here, right? And just do return.
And see it's running.
Okay, thank you.
No problem.
I think that's fine, like if you have like everything specific.
Yeah, I'll just close this quickly.
Into, I think they need to be the fast. Yeah, I think that's why I'm confused with yours, because you